People are usually eager to know the sex of their unborn child. Today, advancements in genetics have made it possible to determine the sex of an unborn child, though the practice is illegal in many countries, including India. However, determining the sex of an unborn child wasn't easy in earlier times, and the mechanism of sex determination puzzled geneticists for a long time. Early research in the field of genetics was carried out using insects. In fact, the development of the chromosomal theory of sex determination took place through cytological studies carried out on insects. Today, sex determination in human beings and several other animals is done with the help of specific chromosomes. These sex-determining chromosomes are called sex chromosomes, while the rest of the chromosomes are called autosomes. For example, human beings have 23 pairs of chromosomes. Of these, just one pair is the sex chromosome, while the remaining 22 pairs are autosomes. The very first indication that sex chromosomes were different from autosomes came from the experiments conducted by German biologist Hermann Henking in 1891. Henking noticed a specific nuclear structure in a few insects during spermatogenesis. However, he also noticed that only 50% of the sperms received this structure. Henking named this structure the X-body. However, he couldn't explain the significance of this structure. Further research by several other scientists led to the conclusion that the structure was actually a chromosome, and they named it the X chromosome. Let's now take a look at a few sex determination systems, which are actually biological systems that help determine the development of sexual characteristics in an organism. In human beings and several other mammals and insects, the sex determination system is of the XXXY type. Human beings have 23 pairs of chromosomes. Of these, the 22 autosomes are identical in both females and males, while the sex chromosomes are different. The sex chromosomes in females are a pair of X chromosomes, while those in males are an X chromosome and a Y chromosome. In other words, the ovum produced in females contains only X chromosome, while in males, two types of gametes, X and Y, are produced during the process of spermatogenesis. Of the total sperms produced during spermatogenesis, 50% carry X chromosomes, while the remaining 50% carry Y chromosomes. And there is an equal probability of fertilization of an ovum with a sperm carrying either of the chromosomes. So, when an ovum carrying the X chromosome unites with a sperm carrying the X chromosome, the resultant XX zygote develops into a female offspring. On the other hand, when an ovum carrying the X chromosome unites with a sperm carrying the Y chromosome, the resultant XY zygote develops into a male offspring. Therefore, it is obvious that the sex of the offspring is determined by the genetic makeup of the sperm. Moreover, it is quite evident that the probability of a pregnant woman delivering either a male or a female offspring is always 50%. Another mechanism of sex determination is the XXXO type, which is applicable to several insects such as grasshoppers, cockroaches, and bugs. In this type of sex determination, females have two identical homologous sex chromosomes designated as XX, while the males have just one sex chromosome designated as XO. Here, the O denotes the absence of a second sex chromosome. Now, when an ovum carrying the X chromosome unites with a sperm carrying the X chromosome, the resultant XX zygote develops into a female offspring. Conversely, when an ovum carrying the X chromosome unites with a sperm carrying no chromosome, the resultant XO zygote develops into a male offspring. Therefore, we have seen that in both the XXXY and the XXXO mechanism of sex determination, it is the genetic makeup of the sperm that determines the sex of the offspring. Such a system of sex determination is known as male heterogamety. However, there are also several organisms in which the mechanism of sex determination is female heterogamety. That is, two different types of gametes pertaining to the sex chromosome are produced by the female. An example of such a sex determination system is the ZWZZ type, and it occurs in birds, reptiles, fish, and some insects like butterflies and moths. In this type of sex determination, females have two different types of sex chromosomes designated as ZW, apart from the autosomes. Males, on the other hand, have identical sex chromosomes designated as ZZ, apart from the autosomes. Now, if an ovum carrying a Z chromosome unites with a sperm carrying the Z chromosome, then a ZZ zygote is formed and it develops into a male offspring. However, if an ovum carrying a W chromosome unites with a sperm carrying the Z chromosome, a ZW zygote is formed and it develops into a female offspring. Another example of female heterogamety is the ZOZZ type of sex determination seen in some butterflies and moths. In this type of sex determination, females have only one sex chromosome designated as ZO. Here, the O denotes the absence of a second sex chromosome. Males, on the other hand, have two identical sex chromosomes designated as ZZ. Therefore, if an ovum carrying the Z chromosome unites with a sperm carrying the Z chromosome, the resultant ZZ zygote develops into a male offspring. However, if an ovum containing no chromosome unites with a sperm carrying the Z chromosome, the resultant ZO zygote develops into a female offspring. Thus, as soon as the zygote is formed, the sex of an organism gets fixed, and it can be determined by analyzing the composition of the zygote.